Common biostimulant products on the market, such as seaweed extracts, humic acids, amino acids, and chitosan, are mostly derived from natural mixtures. Because their raw materials are influenced by factors such as environment, season, and processing methods, the active components are difficult to control, making their functional effects unstable and difficult to reproduce consistently. Homeostatic Systemins is derived from keratin in chicken feathers, offering advantages of low carbon emissions and circular reuse. Through rigorous raw material selection and standardized processing, the consistency of protein structures and stability of functional peptide ratios are ensured. Using a subcritical hydrolysis green process, keratin is efficiently broken down to extract peptide sequences with functional potential. This process is highly eco-friendly, with carbon emissions far lower than those of conventional agricultural input manufacturing. At Biotech, all hydrolyzed peptides are profiled for amino acid sequences using high-resolution mass spectrometry. By integrating AI-driven big data with transcriptome analysis, specific peptides that precisely trigger plant defense and metabolic pathways are identified, leading to the development of Homeostatic Systemins, a precision-induced biostimulant with predictable functions and reproducible effects. Activating the plant's molecular priming mechanism, which enables defense responses to be triggered in advance of stress, is widely recognized as an advanced agricultural strategy. Many biostimulants can also elicit similar responses under certain conditions. However, as natural mixtures with complex compositions and random modes of action, they function much like a black box, with unclear, unstable, and non-standardizable physiological mechanisms. Homeostatic Systemins fundamentally transforms this situation. Instead of relying on random stimuli, it employs well-defined functional peptides with known sequences to deliver precise signaling induction in plants. Analogous to the evolution from traditional broad-spectrum herbal remedies to modern targeted therapies, we achieve three key goals: signal specificity, activating only the intended physiological pathways, avoiding ineffective or negative energy consumption; reproducible effects, ensuring consistent priming effects across different locations, times, and product batches; high efficiency, achieving superior physiological activation with ultra-low dosages compared to traditional mixtures. Homeostatic Systemins provides plants with stable resilience against stress by preventing excessive gene expression and unnecessary energy expenditure. For example, in a maize low-temperature germination trial, untreated seeds under cold stress had to activate more than 800 genes to cope with the environment. In contrast, with Homeostatic Systemins treatment, gene expression was reduced to fewer than 200 genes, limited to the most critical pathways, yet plants still demonstrated strong tolerance to cold stress. This precision induction and optimized energy allocation ensure that crops maintain highly efficient nutrient uptake, metabolism, and growth even under adverse conditions. From rice and coffee to grapes and vegetables, Homeostatic Systemins has consistently delivered higher quality and yield across multi-crop trials worldwide, helping farmers achieve top awards in competitions. In over 2,000 field trials, Homeostatic Systemins showed no negative effects or adverse feedback across different crops, cultivation sites, application rates, methods, or timing. These results fully demonstrate its high stability and broad adaptability in real agricultural environments. Compared with other biostimulant products, its functional consistency stands out as a clear advantage. Furthermore, once the functional peptides in Homeostatic Systemins have completed their signaling and regulatory roles, they are naturally broken down by the plant's metabolic pathways into amino acids, which can then serve as a reusable nitrogen nutrient source. This ensures zero residue and pollution-free safety, perfectly aligning with the dual goals of sustainable agriculture: environmental friendliness and efficient resource utilization. In modern smart agriculture, environmental sensors and digital decision support systems are widely applied for real-time monitoring of climate, soil, water, and pest information, assisting farmers in timely management. However, such sensing systems remain at the stage of external data collection and human response, without the capacity to proactively enhance the plant's own resilience against stress. Homeostatic Systemins introduces an entirely new sensing paradigm, equipping plants with a more sensitive built-in physiological sensor. Its innovation lies in strengthening plants' inherent perception and regulatory systems by using functional peptide signals to mimic endogenous cues and exogenous stimuli, thereby activating early warning responses. This allows plants to initiate a low-cost preparedness strategy before stress arrives, building stress memory and significantly enhancing resilience. By combining external smart agriculture sensors with the internal physiological early warning of homeostatic systemins, crops can not only perceive their environment, but also actively regulate themselves and withstand challenges. This is the next step in sustainable agriculture, and the standard of the future.